Google Hangouts on air. So we are live on the internet. This is so
wonderful.
Yeah. So, this is to keep my committee under control, but also so my mom and dad and some other people can, collaborators can connect remotely. Check out the screencasts. Gary, let me see if I can get you on the screen briefly.
Well, I don't know that that's necessary.
Really? Okay. All right. You're on my screen. I'm watching you.
Okay. So welcome, everyone. Welcome, Ben's mom and dad. I just want to say a couple words because I have had a very bad day and nothing's worked out. So I haven't had much time to prepare. So Ben is the kind of student that everyone would like to have. He's smart, he's creative, and he's interested in a lot of things. And he's now managed to tie those many things together. At least we hope so. Please excuse him if he's a bit bleary-eyed today. He's been working on putting this together day and night for a couple of weeks, so bear with us, please. Go easy on him. Anyway, I'm very happy to be here today. I'm very happy Ben's here today, and I'm very happy to finish this thesis. So take it away, Ben.
So I should add, as his cog-sci advisor, not only is he all of the things that Gary said, but from the moment he walked in, he knew exactly what he wanted to do. And I kept saying, not so easy, you don't have the computational background, you don't have this, you don't have that, but if that's what you want to do, we'll let you do it. And he said, it's either that or that. And here he is.
Great, thanks. Okay, here we are. We finally made it. Title of my talk is Asymmetries and It's time to tell the whole story. We'll see if I can live up to the title. Just wanted to let you know again that we're online, live. This is because Gary, my advisor, is out in Dijon, France. He's joining us from his sabbatical. And he's been kind enough to push me along while he's taking a break out in France. And also just to welcome my dad and my mom and the rest of my family and people connecting remotely. So, yeah, just be aware of that. So, a lot of people, I think, on this campus know me as the guy who loves lateralization. And what's not to love? Lateralization touches on all the things we think make us human, things like fine motor skills or language or things like face perception and emotional processing. But what you might not know is that I'm also the guy who loves interhemispheric coordination and understanding how the hemispheres talk to each other. And one of my goals today is to try and suggest that this coordination matters. It matters, sorry Gary, it matters in I think it matters in the development of lateralization, and I think it matters in basic functions like perception. So in our lab does visual computational vision, and in none of the models do they have hemispheric differences. And that's fine. If you present a stimulus to the left, it will go to the right hemisphere. If you present a stimulus to the right visual field, it will be conveyed initially to the left hemisphere. But if you do something silly like present, right in front of your face, the stimulus will actually get cut in half, a half sent to each hemisphere, which will process that stimulus differently, and will be forced to communicate with the other side at some point to make a decision about what that is or to do something. And I will tell you that that process of communicating is not as simple as you might think. Corpus callosum looks, at the gross level like a single structure, but its fiber makeup is varied, and it seems to have at least two types of transfer. One in primary sensory and motor areas that connect with really thick, fast fibers. It develops early, and you get topographic connectivity just along the midline. And then places like in later visual areas or frontal cortex where you connect with really slow, thin fibers that are very slow to mature, and you get more diffuse connectivity. So how you share information depends on where you are in the visual processing stream. and those two streams do different things. So today I want to talk about 3 projects I did. I hope I make it through all of them. The first is all about lateralization of visual processing. But in the end, I think interhemispheric communication is really important to explain the development of visual asymmetry and visual processing. Then I'll talk about 2 different studies looking at how interhemispheric communication changes with brain size. Everybody thinks, a lot of people think that as brains get bigger, the communication between the hemispheres decreases. And I'm going to look at each of those studies and including looking at development and try and tell a slightly different story. Great. So I'll start by giving a brief introduction into these visual processing asymmetries that I keep alluding to. There's basically two major theories out there trying to describe the data. The first focuses on objects that we're experts for, that we have a lot of visual experience for, so today I'll focus on faces and words. And faces and words both activate preferentially the fusiform gyrus in the ventral temporal area, the same region but in different hemispheres, where the faces will activate right hemisphere more and the visual word forms will activate left hemisphere more. Crucially, kind of this lateralization isn't there from birth, it seems. It comes online around age 5, around the time that visual word learning, that kids are learning to read. And so the theory that's been put forward by Berman and Plough to explain this is that faces and words both require really high acuity central vision. Oops. Oh, it's all over. Let's go down my screen.
What happened?
Gary, we're back.
Yeah, I can see that.
Sweet. Okay. So the theory about why faces and words are lateralized and why that depends on experience is that they share high acuity central vision, but they have very different representations in the brain. So those representations compete. And the best thing the brain can do here is separate across the hemispheres to avoid kind of 2 different representations combating each other. And that language gets left lateralized, or sorry, words get left lateralized to be close to language to make efficient representations there. A kind of whole separate but classic literature shows lateralization and processing of these, at the top, the Nabhan stimuli, these hierarchical letter stimuli, where the left hemisphere seems to preferentially process the smaller local level, and the right hemisphere, the larger global level T. There's also lateralization of spatial frequency grading processing, identifying all the classic kind of psychological experiments that you can do presenting stimuli. And, I'm sorry. So in this model, the encoding of the images is symmetric between the 2 hemispheres. They encode the information the same. What's different is how they extract the information from the images. So the idea is there's a spatial frequency decomposition of the images, and the right hemisphere preferentially accesses low frequency information from those images, and the left hemisphere high frequency information from those images. And that leads to lateralization in response to tasks requiring processing of these stimuli.
Ben, if it matters to you, I can't see the slides anymore.
Okay, thanks for letting me know.
You could just point your camera at the board if you want.
That wouldn't go so well for my on-air hangout. I want to make this happen, Gary. Let's do it the right way. That should work better, right?
Yep.
Okay, let's just not crash the Hangout again. So the theory I'm going to talk about today, our hypothesis with Gary, all this work is with Gary, is that the right hemisphere, is that there's a specific neuroanatomical mechanism that lateralizes, that's different between the left and right hemispheres from development. And that representations of things like faces, I can't move, sorry Gary, faces, spatial frequency gratings, large hierarchical letters, and contours, just general contours. They all have one particular type of representation that neuroanatomical setup of the right hemisphere preferentially processes. And words have a different kind of spatial representation, spatial information, and that's preferentially processed by the left hemisphere. Our view is that it's something called long-range lateral connections, and I'll explain what that is, are what's different between the hemispheres. And we have a neurocomputational model trying to show that an asymmetry in these long-range lateral connections can affect the processing of each of those stimuli. So I'll start first by talking about this model. And I'll start first on that talking about connectivity. Because I think most people will be familiar with white matter connectivity between cortical areas. That's just under the surface of just under the cortical surface. This is like feed forward and feed back inter-area connectivity. But the cortex is a sheet, and you can inflate it and then flatten it into a sheet. It's just a little bit thick. And if you inject a special kind of dye into the cortex, you'll get this kind of patchy labeling.
Ouch.
Ouch? Did you say ouch? All right. I'm not even going to argue with you, even though I'm tempted to. So here's the injection point. You're zooming in, and you see this patchy labeling. And what you'll see is a dark patch at the injection point, suggesting highly connected local set of neurons, like a cortical column, large lateral column. But you'll also see sparse, so this is all cortex, but only these are labeled, suggest sparse interconnections between columns through the gray matter itself, not out into the white matter, but through the gray matter. These are long-range lateral connections. And so if you map that onto a play, kind of retinotopic visual cortex, where each of those circles represents, a column with that receptive field, you can see that, your red injection point has some neighbors that it connects to and it gets information from. And we can turn this into a really simple neural network. The idea is that the red node just gets activation from itself and its neighbors, and what it's trying to do is, let's say the stimulus is really weak and you're not sure what it is at this location, what's going on, what orientation is there, let's say. You can consult your neighbors and you can ask them, hey, well, I'm not sure what I saw, what did you see, right? And through experience, through learning, you can say, okay, I tend to see things that are curved or that are straight, but I don't see jagged things, right? So if your neighbors report something that's consistent with the curve, you're like, I'm more confident, my orientation was right. If your neighbors report something different, you're like, there's no consistency. So the idea is the neural network's just finding contours or correlations amongst its neighbors to find consistent features. This neural network is just like the first one, except it takes that activation with neighbor information, and it just uses it to recompute the original image. It's called an autoencoder, and this can be used to learn the weights feeding into this unit to figure out how do I best use my neighbor information to learn statistics of the images. And so this is our model. I showed you one hidden unit there. Oh boy. See if that can go away. Showed you one hidden unit here, but I have one of these for every pixel in that image, centered on that pixel. And the parameters of the model are, I can choose how wide to spread those connections. And the question we're going to ask is, how does the spread of those connections affect what what neighbor information I use, what features I determine, are relevant, given far away neighbors or close neighbors. And so this is the asymmetry that we postulate to be in visual cortex, that the left hemisphere connects these long-range lateral connections to more distant neighbors, and the right hemisphere to more close-by neighbors. And you can see the Gaussian distribution will select connections from is just more narrow in the right, more wide in the left. And we're going to create this neural network and see how it affects frequency processing. Okay, so this is the basic training paradigm. Is we have 850 of these units. Now I'm showing you 2. There's a lot more. You center a Gaussian on each unit, and you randomly select connections. You select a set of images that you're going to train this network to just reproduce. So initially, the network starts, and it reproduces something that's very noisy. And so you change the weights. There's some algorithm to change the weights such that the next time you propagate the information forward, it gets a little bit better, but not quite good enough. And then finally, with any luck, I can do it one more time, and the image will look pretty good. And so we're going to say that's enough. I've trained the neural network. It's learned neighbor information based on that relatively wide connectivity pattern. And then I can do two types of analyses to understand what this network's doing. First, I can take the output image, and it's not a perfect replication of the input. And I can see how well did it reproduce that image, right? But specifically, I want to know how well did it reproduce that image in the low frequency information and in the high frequency information. And I want to see, as I vary those connections, if it affects where the error is located in the image. Second, I can take these internal representations biased by the neighbor information, and I can do behavioral tasks with them, just like you put people through. Tell me if there's a T in that image. You can make a neural network take these biased versions of these images and do that same task and see if the neural networks with different connectivity patterns perform differently. And so that's exactly what I'm going to do now. The first task we tested the network on is this lateralized presentation. Target letter T, just press a button if you see a target T. It's on the left. That'll be initially processed by the right hemisphere. If it appears in the right visual field, it'll be preferentially processed initially processed in the left hemisphere. And with the same stimulus, if you see different reaction times if it's presented left or right, that's some indication that the hemispheres are doing different things. And so the task here is just press a button if you see a target letter T, target level H. Doesn't matter if it's big or small, just press the button. And what you find in the behavior is if the target is big, the global level, you'll see faster reaction times as it's presented to the right hemisphere to the left visual field, as shown here. If the target letter is at the smaller level, you'll get faster reaction times if it's presented to the left hemisphere or right visual field, shown here. And so this is what we do with our neural network. We train the network. We sample connections differently, wide in the left, narrow in the right. We just train on a whole set of these Navant figures. It doesn't have any tasks but to reproduce the image. It doesn't do it perfectly, but we train it to do it. And then we record these hidden unit activations, and in a separate network, we train it to do the behavioral task. Press output 1 if you see a T, output 0 if you don't see a T, and we see which network does better for which stimulus, how much error is in that network. And so here's the behavioral data of the humans, and here's the output from the network. You can see that it replicates the same hemisphere by target level as you see in the behavior. And so we see an association between these connectivity differences in local and global processing. On the same task, instead of taking the hidden units, again, I can take the output images and I can analyze the spatial frequency, the error in these images in terms of spatial frequencies, and just compare which network is better at representing high and low frequencies. And so that's what I'm plotting here. Above, I just, to compare that, I just subtract right and left hemisphere. So above the line means the right hemisphere is doing a better job of representing information, spatial frequencies. And you can see the right hemisphere generally represents information in lower frequencies better. our right hemisphere network. And the left hemisphere network tends to represent information in the higher frequencies better. And if I just filter those images based on the frequencies there, you can see that the right hemisphere network seems to be doing a better job representing frequencies that are more at the global target level. And the left hemisphere network is better representing information at frequencies that are more like the local level. So this may be driving some of that behavioral differences. I can do the same thing on different image sets, so I can train these networks on faces and analyze the spatial frequency outputs of that, and I get a similar interaction where the right hemisphere is better for lower frequencies, left hemisphere network for higher frequencies. I can do it on a whole data set of natural images, whether they're looking normal to us or whether I've warped them to be more like how cortex is. So cortex over-represents things at the fovea, and so you get this log polar stretched representation. Things at the center are quite stretched. If I use that representation in these networks, I also get these same spatial frequency patterns. So suggesting that the connectivity is driving encoding differences between the hemispheres. Third thing I did with this model is rather than selecting the connections and seeing what spatial frequency information is used, I actually trained two identical networks on different spatial frequencies and allow them to choose the connections that they can use best to encode that information. So this follows a developmental paradigm where early in development, visual acuity is poor and contrast sensitivity is poor. It improves over time. There are data showing that the right hemisphere begins maturing earlier than the left. So the right hemisphere will be maturing with more low contrast, poor acuity images than the left hemisphere. And then these patchy connections, it's known that they mature by strengthening the connections, but also eliminating irrelevant connections. And so the right hemisphere will prune connections under blurrier, lower spatial frequency. Okay, so the way I set this up is I start the right and left hemisphere networks again with the same exact same connectivity pattern. So I sample random connections once and I duplicate them for the two networks. I'm just showing one unit here. Again, there's 850. Then I train on natural images with the right hemisphere network receiving lower frequency blurry images. And while training and adjusting the weights, I also remove the weakest connections. And at the end, through that process, the two may wind up with different sets of connections. And so the only asymmetry here is on the inputs that they're trained on, not on the original setup of the networks. Here's just the schematized, oh boy, version. That's really blurry. But this is a blurry F, and it gets less blurry over time. And you can just see that the right hemisphere's blurred Fs are more blurred than the left hemispheres. And in the end, they both train to some criterion on the full fidelity images. After I do that training, I just take these units with the remaining connections, and I put each one at the center, and I just compile the distribution of connections. And I want to see if the connections are different between the networks. And so here's the right, here's the left. It's hard to see what the difference is, so I can just subtract them again. And the warmer colors mean more in the right hemisphere network. And it's showing that the right hemisphere network trained on the lower frequency images, again, prefers the closer together connections. So it kind of validates the previous result. So just to summarize what I'm showing in this network, the right hemisphere seems to be specialized for encoding local spatial relationships. Yeah, I'll actually go into a little bit more to explain. Yes?
I don't know if it's relevant, but Marlene says she can't see the slides.
Okay.
I don't know if there's anything you can do about that.
I don't think so.
Okay.
Can you apologize to Marlene for me?
Well, so John and Marlene both emailed me. I just noticed.
Mm-hmm.
And they said the slides are minimized in the bottom right. So I think if they click on those,
Yeah. Exactly. Tell them to click on me. Okay. So thanks for that. And Marlene, if you're listening, click on the slides in the little bottom right corner. Okay. So the right hemisphere is specialized for encoding local spatial relationships. And it's specialized because it has shorter long-range lateral connections than the left hemisphere. And that this connection asymmetry can come from normal human development, where the right hemisphere tends to start maturing earlier under poor visual acuity and development using connection pruning. Just to explain what I mean by, oh boy, by local spatial relationships, what I've done is I've taken a natural image or a face, right? And I've taken a pixel, and I just correlate that pixel across all the natural images or all the faces with its neighbor, or with its more distant neighbor, or with its more distant neighbor. And you can see that as you correlate the pixel information from one neighbor to nearest neighbor or farther away, the correlation decreases. There's a high correlation in spatial neighbors and low correlation in distant neighbors. And let's just say that that white spot is broader, a broader Gaussian than what you're seeing here. So there's a lot of spatial correlations in natural images and faces. However, if you look at words, what you see is there's actually a line here, and it's pretty dull but pretty widespread. And what it's saying is that there's not a lot of spatial correlation in information in visual word forms for neighbors, whether you're close by or more distant. So what I'm suggesting is that nearby connections will pick up more spatial correlations than more distant connections for natural images and faces. But for words, whether you're close by or farther away, there aren't a lot of spatial correlations to pick up, and so it won't matter so much. And that's why you get this right hemisphere preference for things like faces and contours and natural images, things that have smooth shapes. They'll pick up that information well. And the left hemisphere's wider connections, it won't matter as much, and so the words will be lateralized to the left. Okay. Going back to the developmental model, our developmental model shows asymmetry in the connections, but actually it shows weaker lateralization than the original networks we had. And connection asymmetry develops early by 15 months of age. That's when pruning of these connections is done. And we did it without hemispheric competition. But those developmental data I talked about in the beginning suggest that this lateralization comes online from 3 to 5 years and keeps developing through 14 years old. So how to resolve it? A potential solution, there are two that are possible. One is that I could just make the differences between the left hemisphere and right hemisphere images bigger, so blurrier, even blurrier right, even stronger full fidelity left. Or it could be that interhemispheric connections actually cause competition, as postulated by Berman and Plout. And I think this is a more likely solution. Yeah, just given some other data. And so the hypothesis is that an asymmetry would emerge in the connectivity between 3 and 15 months when those connections, long-range lateral connections, are being pruned. After 15 months, further visual experience will actually suppress asymmetries as the two independent hemispheres try to represent information fully. But then as callosal fibers start to mature, you'll get a competition between the hemispheres, and you'll start to get a bias in word learning, pushing words to the left hemisphere, faces to the right hemisphere. It's quite a story. The question is are there any mechanisms that would actually make this more plausible? And so what I'm going to talk about next is some mechanisms that I think make this more plausible, and how. Everything okay? Is Grant still there?
I'm still here, but everybody online can only see my face.
Okay. Well, this is. I hope my mom is getting to know you. I don't think you two have ever met before, right? So maybe write her a message on a piece of paper and just flash it to her and say hi. Hi, Mom. So I'm going to tell you about the corpus callosum for the rest of the time. And I'm going to talk about two ways people think the corpus callosum prevents communication in large brain species like humans. And in doing so, I'm going to talk a little bit about callosal development, how I think this whole story backs up that kind of fantastical story I was just trying to tell. So,
okay, and what I'm trying to do is I'm just doing a screen share, so hopefully people that are watching me will now see the slides.
Cool. So the question is, are the hemispheres interdependent or independent? Because it seems at some level like they're independent. If before surgery to test, well, you can inject into someone's carotid artery a chemical that will essentially turn off one hemisphere. And when you do this, if you turn off their left hemisphere, they won't speak, right? They'll lose the function to speak usually, but they won't freak out. They won't say, half of me is missing, or everything looks blue and checkered green. Nothing like that. Things just seem sort of regular. And at the same token, if you cut the corpus callosum in adults, and then you ask after the surgery, long after the surgery, how are things going? And so there's a YouTube video you can check this out on where Alan Alda asks this colostomy patient, JW, do you feel any different now without a corpus callosum when you think? He says, no, I just got a backup brain now. So these things seem to suggest that the hemispheres are somewhat independent. And it also seems like lateralization may be related, and yeah, kind of made a conceptual jump here, but let's just go with it. It also seems like brain size may be related to lateralization because we have big brains. We think those make us human. Lateralization touches on all the things we think make us human, so probably they're related to each other. And there's two major papers that look at this. One that says that, yeah, because we have big brains, the delays between the hemispheres are really long, and it doesn't pay off to try and communicate between them. Better to communicate within a hemisphere where delays are faster. Yep, that the number of fibers that connect across the hemisphere are proportionally fewer when compared to intrahemispheric connections in bigger brains. And so each of these will lead to independence, and that's what's driving lateralization. And these are functions of brain size. So we have big brains, we have really independent hemispheres, and we're really lateralized. I think this is kind of in the wrong direction, that we're distracting ourselves with these hypotheses. We see anatomical and functional asymmetries in large and small brains. Humans that don't have a corpus callosum, so either they have really, really long connections, really long delays, or just very, very few fibers, they don't show greater lateralization, as you would expect in this theory. And although delays, like conduction delays, decrease with development, lateralization increases during development in language, in handedness, in vision. And finally, when you look at functional connectivity, so just put someone in a scanner and then brain activity evolve with no particular task, and then you correlate, changes in hemodynamic response, changes in blood flow, essentially. From one region of the brain to another, you always see the greatest correlation between the same area in the corpus callosum, suggesting that there's potentially some special relationship there. Okay. So in this first model, our analysis of the data showed that the difference in the models is not a difference in interhemispheric communication. It's actually a difference in the onset to interhemispheric communication. Then when you have longer or shorter delays between the hemispheres than within the hemispheres, you will still communicate between the hemispheres equally. It will just take a little bit of extra time for that communication to begin. So that's actually different than what was originally stated in the paper, that it fundamentally changes the communication. It doesn't. It just delays it slightly. And I don't think delay really matters. That onset delay really matters. First, the magnitude of the delays that they had in this model are on the order of 10 times, but in the real brain, more like 1 to 3 times if I had to estimate. The corpus callosum contains a distribution of delays, and it's possible that these very fast delays can mitigate the effect of an onset difference between the hemispheres. And third, things on the order of 5 to 20 millisecond delay only matter if there's zero prediction. Zero prediction, like your brain's starting from scratch, and there's no temporal correlations. And I don't think that's what the brain does. And so I don't think these magnitudes are actually very relevant to computation whatsoever. I think that the issue is that these conduction delays correlate with things that really do matter. So the longer the connection, the longer the delay, and the greater the white matter volume. And if you look at how white matter scales with brain volume, you'll see that as brains get bigger, the proportion of white matter increases to the point where at some point, if you had a big enough brain, it would be 99.9% white matter, right? This is untenable. And so one reason you would reduce the number of long distance connections is because you can't do that. You don't want to do that. Second is metabolism. The brain uses 20% of our body's energy, and long distance connections, especially if they're unmyelinated, can be very expensive. The third thing I want to focus on is variability in the conduction delays. So this is the idea that the longer the conduction delay, the more chance there is that it won't arrive at the same time every time. And variability in conduction delays do exist, but I haven't really seen them addressed in the cognitive literature. And the time at which they exist most is early in development, where prenatally, you see that in the corpus callosum, most of the fibers are really, really thin and unmyelinated. So myelinated fibers are black, unmyelinated are white. And over development, those axons get bigger, thicker, and start being myelinated. And actually, these small unmyelinated axons in the red box there have reliable delays, up to 20% variability in their time. And so the argument I want to make is that this affects overdevelopment, how much you will want to use your callosal delays. You won't want to use very unreliable delays. And as they get more reliable through development, you'll use them more. And I don't really have time to go into why, but I do believe the degree of this effect may be greatest in humans, where we have a trade-off between big brains with long fibers, but also high compression of our brains. We're kind of at the limit of how big our brains can be and still give birth successfully. So the question in the modeling I did is, what are the effects of this delay-dependent variability? And so I used a neural network that had, again, delays on connections, short delays within a hemisphere, long delays across the hemispheres, but the longer delays had more variability on the connection. And what I found is that when the delay is reliable, like, there's no interruption in long-distance communication. There's just a slight delay to its onset. But for when the delays are unreliable, then you get a bias for intrahemispheric processing, because you've got essentially noise and things that you're trying to learn on those long-distance connections. As much. And then as the variability is reduced over development due to the thickening of those axons and myelination, you'll have greater and greater interhemispheric collaboration. And if you note that, again, the lateralization of language, handedness, visual processing increase with age, then you get a coordination. Then you can see that the two seem to develop in tandem, right? You get more interhemispheric coordination with age, more lateral. So it doesn't fit this hypothesis either that coordination and lateralization go in opposite directions. In fact, I think collaboration is strong and may enhance the lateralization. At least that's correlation, not causation yet. The last part here is I wanted to examine this claim that there are fewer interhemispheric connections in larger brain species. And to do this, we need to understand a little bit about allometry, and I'm in the room with experts on allometry, so I can do my best. So the idea here is that there's a large kind of difference in magnitude of mammalian brains, But big brains are not just little brains. You don't just take a little brain and then blow it up like an image and it's a big brain. The architecture of the brain changes as it gets bigger to compensate for things like delays, volume, from metabolism. So for example, in cortical folding, if you had a sphere, right, and you just compared how the surface area of a sphere should change with volume, should scale as the 2/3 of the volume. So volume goes as r cubed, surface area goes as r squared, and so the comparison of the two should be to the 2/3 power. In actual brains, it goes closer to 8/9 power, suggesting we have more surface area for the volume than you would expect if it was just smooth in a sphere. And this is, in some sense, a description of the fact that smaller brains have more smooth surfaces, and larger brains have more and more convoluted surfaces. This is just an example of how brain architecture changes with brain size. And there are three basic relationships that can happen, or power law relationships. So some quantity on the x-axis, like brain size, brain volume, and some other quantity on the y-axis are related by a power law. It can be, the power can be less than 1, which suggests what's on the x-axis is growing larger, faster than what's on the y-axis. It can scale with it isometrically, so x and y-axis are scaling at the same rate, or the y-axis is scaling faster, the power is greater than 1. If you plot these on a log-log axis, you'll find a line that's either sublinear, so slope less than 1, slope greater than 1. And so you can just use linear regression in most cases to analyze these things. This is to say that for some people believe that this controls all quantities in the brain, including microscopic quantities like neuron density, the macroscopic quantities like white matter volume, or even the way that areas connect to each other in the brain. So in this original study, 11 primate species, full MRI volumes were taken for 11 primate species. And what Rilling and Ninsel, the authors of this paper, aimed was to compare the total number of fibers in the brain versus the total number of callosal fibers, and to see how this scales with brain size. What they estimated was the surface area of the gray matter as a kind of rough proxy for the number of neurons and the number of connections, and the callosal surface area as a proxy for the number of callosal connections. That's what's available through the MRI images. And when they plotted these against each other on the x and y axes, they found that there was an allometric relationship, that on a log-log axis it was well described by a line, and that the power was less than 1. So what's on the x-axis scales faster than on the y, and that's the gray matter. That's the total connectivity versus the callosal connectivity. And so they use this as the argument that there's proportionally fewer fibers in the corpus callosum. With larger brain size, they're more independent, and that's going to lead to lateralization. I think there's two major issues with this study. First is that the missing quantities that you need to estimate connections from surface areas, so like the neuron density or callosal axon density, those also change with brain size, and so you can't just make the assumption using the proxy. I think you actually need to plug them in and estimate. The second is that their result was confounded with how inter-area connections scale. So the idea is that the corpus callosum is largely homotophic. It connects an area in the left hemisphere with an area on the right hemisphere, and vice versa, and generally not much at all with any other. Mostly just one, regardless of brain size. On the other hand, with intrahemispheric connections, the number of areas that you're connecting to increases with brain size to keep kind of network computational properties. And so without accounting for this, you might expect some different scaling of the connections. Let me go into a little bit more detail with that. So I have a small brain that has 8 cortical areas per hemisphere, a large brain with 16 per hemisphere, and the smaller brain connects to 4. One area will connect to 4 other areas, but in the larger brain, one area will connect to 8 other areas. So as the brain got bigger, there were more total connections. Only one of them is an interhemispheric connection. And so it looks like interhemispheric connections are proportionally being reduced with brain size. But if you take any individual intrahemispheric connection, the same thing's happening. So for example, from B1 to B2, that connection would be going from 1/4 to 1/8 as well. So this isn't a property of interhemispheric connectivity, it's just a property of how the brain scales. And it's just not the right measure of looking at Great way. Harry, please.
Sorry.
You having a Snickers again?
Nope.
Oh, well, that's too bad.
I had to add some red wine to my chili.
Okay. So I think the right thing to compare is what we do in DTI, where we compare the number of fibers in a fiber tract compared to others. And see if one of these fiber connections, the interhemispheric fiber tract versus the average intrahemispheric fiber tract, if those have different numbers of connections within them, compare that value, and that will tell us if there's selective reduction of different fibers. I may cut you off, Gary. Okay, so I did two things. First, I just tried to estimate the total number of fibers and total number of callosal fibers, and then I tried comparing that to the scaling of the areas instead. And so here's the equations. They're really simple. The number of callosal connections is that callosal surface area times the axon density. The number of white matter connections is the total number of neurons times the percent that send projections into the white matter. And the total number of neurons is just the gray matter volume times the neuron density by volume. Most of these are available in the literature, and I can plug them in. The one that's not is the density of the corpus callosum, but a recent paper has published 6 species showing neuron density, and so I grabbed those values, tried an allometric regression. It looks relatively linear. Humans are way out here. We have way bigger brains than any of the species used in this regression, so that kind of extrapolation is dangerous. So I take data from the literature. I massaged it into a form to make it comparable to the animal data, and it fell right along the regression line. So I think it's a valid regression to use. And it's worth mentioning that the scaling of the callosal density goes as the minus 0.28 power, suggesting that as brains get bigger, the corpus callosum density decreases. The fibers get bigger, more myelinated, and so you have fewer fibers per unit surface area. When I plugged it in, I got a much lower power. So this is saying that, yeah, the intrahemispheric connections scale way faster than those interhemispheric connections. Not as mildly as you were reporting before, but actually much more severely. So why is that? Like, that says these hemispheres, under their logic, are even more independent than ever expected. In fact, I think more than is really tenable. And so how much of this can be explained by this compound with inter-area connectivity? So all I did is do a ratio, callosal inter-area connectivity versus intrahemispheric inter-area connectivity. Compare that to the ratio of callosal fibers to total fibers. And the expectation is that if this explains this, then this should scale with isometry. This should look the same. And that's essentially what I got. I got to the power 1.04. So very close to isometry, suggesting that these two quantities are highly interrelated. In fact, essentially all the variance of one can be explained by the other. If I can use just a bit of algebra to take that result and compare the average number of connections within a callosal inter-area connection versus intrahemispheric inter-area connection, and I find that there's about 4.3 times more fibers, the inter-area connections across the callosum than within a hemisphere, suggesting that those connections are very important. And so just to summarize, Rilling and Insel suggest found an exponent of 0.88. So the proportion of interhemispheric fibers mildly decreases with brain size. They interpret this to suggest there's a functional difference, functional decrease in flow of communication with brain size. I found that the proportion of interhemispheric fibers drastically decreases with brain size, but that this suggests no functional decrease in callosal communication. And instead, callosal fiber bundles may be much stronger than the average intrahemispheric fiber bundle. So there's certainly no evidence of hemispheric independence in humans, no relationship between the independence and lateralization. So again, I don't think this holds up under scrutiny either. Instead, I'm stuck on this idea that collaboration is strong in humans, just as strong as in other species, and may enhance lateralization through development. Okay. So to summarize, I suggested that development and interhemispheric communication drive differences that lead to visual asymmetries. I offered a mechanism for where a developmental bias towards more local circuits can come from. That's conduction delay variability. I stated that conduction delay magnitude likely does not cause independence, and that independence is unlikely to cause lateralization anyway. And I showed that callosal connectivity is robust across species, including humans, with evidence of a special role. And so our view, what seems kind of obvious to me, is that independence is like being totally self-reliant, right? Like when you're out in the desert all by yourself, you can't choose to specialize. You know, am I going to carry the food or am I going to carry the water, right? You got to carry everything. You got to solve every problem yourself. You're all alone. But when you have others and you communicate with them, you work with them, you collaborate, then someone can, you know, write the computer programs and someone else can do the farming, and we're going to share our resources together. And this is the way I think of lateralization and how the brain works. Sharing of resources allows, enables lateralization, specialization, and name processing. Okay, and with that, I just want to thank a few people. My one chance to thank people. I wanted to thank Gary and our lab for putting up with my neuroscience interests. Even though we sit in computer science and there are posters of machine learning all around. Wanted to thank my committee for being here and for reading through the dissertation. Particularly to Katerina for spending all the time with me in her classes in the lab and really getting me interested in a lot of these issues. To thank Pascal and all the people from CARTA who have been phenomenal. You know how instrumental CARTA has been in my experience in graduate school. And just want to thank you for those experiences. And also to the Temporal Dynamics of Learning Center and Penn. These are kind of my closest collaborators, people I see 4 or 5 times a year, and who I present most of this stuff to most frequently, and so give me the most feedback of anybody. To my collaborator, Janet Shao, and to Gary, who did a lot of the differential encoding work with me, and to some graduate students who have really participated in my research, like Kari Hansen from Katarina's lab, Eva Dundas, a graduate student with Marlene Berman, Kaya Jasinska, who works with the ISLC, which is a super organization of the TDLC, and Gabriela Musacchia, who's a graduate student in TDLC. And of course, my mom and my dad and my loved ones, don't want to forget them. And of course, my funding sources, CARTA and the NSF. And thank you for your time. Okay, so in some sense, we have time for questions, and in some sense, we're over time. So if people need to leave, totally understand, but I'm happy to take questions if you have time to do so.
I have 2 questions.
Yes.
So for your first part, so first is when you train the neural network, this is DPF or?
The neural network's trained using basic back propagation.
It's kind of along the line of DPF, right?
Okay. Okay.
So it's like when you train the right and the left hemisphere, you present with different signal it's like either blurs. It sounds like it will be more direct if you can learn, just present with the same image, but based
Right.
upon different kind of time response of these to automatically learn
Right.
the intrinsic structure.
Right. So for the first two studies, that's exactly what we did. We just had the connectivity difference, and then we showed the asymmetry was in the connectivity difference, and I showed them the same patterns, the same images. It was only in the third study where I showed different images and then allowed the connectivity to change just to see if that produced the same association between short connections and low frequency information to corroborate that result, because that was a somewhat unexpected result.
So then in the difference in the connectivity. So, so how do you specify why?
Yeah. So let me go back if I can. Okay. So this is the developmental study. Let's see if I can go through this. So in the developmental study, this is the only one where I started with symmetric networks, but I presented them with different information. And what I did is, while I was training, I trained for a few epochs, and then I just look at the weights, and the smallest percentage of weights, I just remove those connections, and then I continue doing that through a number of epochs, all the way until I got to the final epoch where I just trained to some error criterion. Does that answer your question? Or?
Yes, that we can. Another question is
Sure.
about if you're showing those response correlation between words and the images. So for words, there's no scale difference, but for images, there's scale variance. So did you?
Yeah. So for the natural images, I didn't do anything with the scale. So let me see if I can just bring that slide up so everybody understands what we're talking about. So you're talking about, I think, this slide.
Yes.
Yeah. So with words, they were all the same font, and I was just shifting fixations and correlating, essentially. For faces, the faces are actually highly co-registered, so there isn't any scale variation there. For the natural images, there's all scales. And so the fact that you get similar statistics in the correlations here and here, I think speaks to the fact that you just get gradual variations. And I don't think you get gradual variations that are in common amongst the images. But I think if the faces did have more scale variance, you might see less correlation, but I think you still see the same general pattern. Yeah. Pascal?
So you obviously worked on vision. What is known about lateralization in congenitally blind people?
In terms of vision?
No, in terms of lateralization.
Or sorry, I mean, so some people think of lateralization as a single thing, and I probably talked about it a lot that way today. But each system, I don't think we have strong ideas about interrelationships. So for language lateralization in the blind, I know in the deaf it looks largely the same. For language for blind, I'm not exactly sure. But for one thing that would be challenging is for blind. There's I believe there's evidence of using, for example, I know area MT, motion area, gets used for auditory information, and so I'm not sure if the lateralization in visual areas would still be supported or not. Given the fact that we and Berman and Plout are arguing that it's activity dependent, experience dependent, then I would not expect it to necessarily show up unless blind were doing something that had competition between representation. Yeah.
Do we go first with?
Let
That's my question.
That's the answer. So we'll finish with questions from the audience before we start as a committee. So if there are some questions, then we'll say not. There's going to be a little bit of party in the grad lounge.
Okay.
And hopefully things won't take too long. So within reasonable amount of time. Are there any more questions? Okay. So then I thank you. Will you stay? we'll see you all in a little bit. It's not quite my shoulder.
Gary, are you still there?
I'm just falling apart.
So Ben,
Something.
I'm going to try and get Gary up on the screen.
So, but the rest of the hangout.
It's off there, yeah.
You're right.
But give me.
Ginny, why don't you move, because my neck is going to break if I have to keep turning around to see you. So,
Also,
Katerina, let him.
committee members present on the.
No, that's,
Just asking.
no.
so Ben,
No,
Just Gary. Well, there's only one left.
to end the
Cut everybody
live
else off.
broadcast for this,
What am I trying to do? I'm trying
As
to
soon as he
get
gets
Gary.
there, you can.
what she should do is
That's
end
one
the live
paragraph.
broadcast and then hang out with me directly.
I think Gary should be. Gary, are you still there?